Hi, welcome to Chapter 3. This is the first of what will probably be 2 screencasts on cells. Cells, as you can see from this image right here, are totally cool, beautiful things. There's really no information in this image. I just love it because it's pretty and fluorescent. The outside of the cell here is in red. Blue is all of the chromosomes. That is a fluorescent dye called DAPI. I used to know what DAPI stood for, I don't anymore. And the red is the mitotic spindle. So this is a cell that looks to be in about metaphase, getting ready to go through cellular division. Or getting ready to actually do the dividing. Let's see, there's no new information on this page. If you click here, it takes you to a cool little website where you can look at different sizes of different things. You basically zoom in past a coffee bean, a grain of rice, and then big cells, small cells, all the way down to a carbon atom. It's kind of cool. But this is what we really need to talk about, which are cells. Before we go into all the nitty-gritty, I kind of want to give you my thumbnail sketch of how I view cells working. And I use computers as an analogy, right? So we've got these 3 parts of the cell right here: the nucleus. All of this stuff is the cytoplasm, so all of the organelles are found within the cytoplasm. And then the physical external boundary of the cell is the plasma membrane. So if you think of a computer, the nucleus is kind of like the hard drive. That is where all of the software is stored. So all of the information that is required to manufacture all of the proteins that do the doing inside of a cell. Like if you remember that crazy chart from Chapter 2 with all the enzymes, right? The blueprints or the recipes for all of those enzymes is right there in the DNA that's stored in the nucleus. And the cool thing about the way human cells work is basically every cell has the same hard drive. So every cell in your body has all of the software needed to run any cell type in the body. It's just that what makes one cell different from another cell is which genes it uses, or which parts of the DNA it uses to make the proteins it needs, which is kind of cool. And then to extend the computer analogy, if the nucleus is the hard drive, the cytoplasm is your desktop. That is whatever you are working on your computer at that moment, whatever programs you have open, browser, whatever it is. Most of what happens inside of a cell happens in the cytoplasm. So all the biochemistry, synthesis and degradation reactions, everything that goes on, all the big biochemical action takes place in the cytoplasm. Then you have the plasma membrane, which I kind of think of as the outside of the computer, which also in my mind includes the keyboard, your USB ports, power ports, right? Everything that gets into or out of the cell has to cross the cell membrane. But the cell membrane is also how the cell communicates with with its external environment. So there are proteins stuck in the surface of the cell membrane that act like keys on a keyboard, and developmental cues or other sorts of cues bump into the outside of the cell, activate these proteins, which then causes stuff to happen on the inside of the cell. Just like when you press the keys on the keyboard or click the mouse, stuff happens on the screen. Same thing with cells. Stuff bumps into the membrane, or the membrane bumps into stuff, and the cell can interpret and respond to all of those external stimuli. Speaking then of the plasma membrane, this is where we are going to start. So here is a plasma membrane. It is composed, as we said in Chapter 2, primarily of phospholipids, and these are all of your phospholipids here. Then embedded in the membrane are proteins. You'll notice some of the proteins are just on the inside, maybe some of them are just on the outside, and some of them go the whole way through. Then you have also just a smattering, a little bit of cholesterol That is used to regulate how stiff the membrane is. We don't really need to get into it because it doesn't affect the way we think of the body, but just remember that the cholesterol is there. We are going to start by talking about the phospholipids and then move on to some of the things that proteins do. So first up is our phospholipid. So as you recall from chapter 2, it is a glycerol head group. So those 3 carbons right there are your glycerol, and then 2 fatty acid tails. And instead of a 3rd fatty acid tail, there's that phosphate group on the end, which makes that end polar and this end nonpolar. So the way it works in your cells is that the phospholipids form a bilayer where the hydrophilic polar heads are facing outside. So this would be the outer surface of the cell. This would be the inner surface of the cell. You want to remember that all the compartments of your body, inside your cell, outside your cell, in your bloodstream, wherever you are, it's a watery environment. So this is watery fluid out here. This is watery fluid in there. So what you basically have is a hydrophobic barrier, or an oily barrier in between 2 watery environments, your intracellular cytoplasm and your extracellular fluid. It is the phospholipid bilayer that sets the baseline permeability for the cell, by which I mean if something can cross the phospholipid bilayer, then the cell can't control whether or not it crosses it. If something can't cross the phospholipid bilayer, and it needs to, then the cell has to build machinery to transport those things into and out of the cell, and that also means the cell can control these things that go in and go out. So because the phospholipid bilayer is a lipid bilayer, it is hydrophobic molecules that pass through it. These are things like gases, so conveniently for us, CO2 and oxygen diffuse right into and out of our cells, and any other non-polar molecule. I'm having trouble thinking of any. Your steroid hormones, because they are derived from cholesterol, pass right through the membrane. Things that do not pass through the membrane are anything with a full charge, like an ion, that should be an S, and polar molecules like glucose. Water happens to be polar, so should it pass through the membrane? If you're thinking, no, I don't blame you, but water can. This is a weird thing about the phospholipid bilayer. Physics predict that water should be able to pass through it a little bit because it's not a hard sort of anatomical barrier, and there's room for water to sneak in between the hydrophobic tails, and with enough pressure water could squirt through, but the membrane is still more permeable than we would necessarily predict. So it's one of those things that we don't really understand. But what that means for us is that your cells cannot control whether water stays inside the cell or outside the cell. Because it can pass through the phospholipid bilayer, your body cannot control where it goes. This is going to become important later. So that's the phospholipid bilayer, which we'll come back to when we talk about transport and how stuff gets into and out of the cell. We're going to shift gears for a second and talk about the proteins and some of the things that proteins do. As it says up here, there are transport proteins and receptors. So this thing right here that says channel, it's basically a big tube that's stuck in the membrane, and things that can't pass through the phospholipid bilayer, like sodium and potassium, can pass through a channel. I don't see anything that looks exactly like a receptor, but for receptors, these are proteins that are stuck in the membrane that, as I said, they're kind of like buttons on your keyboard. you push on the outside of the receptor, or if like a neurotransmitter hits a receptor on the outside of a neuron, that's going to cause changes to happen on the inside of a neuron. The other analogy I use is the dial on an analog watch, if anybody still has one, right? You twist the dial and you can change the time on the watch. That's the way receptors work. Things bump into the outside of the cell, signaling molecules that then cause changes to happen on the inside of the cell. This right here, cell-to-cell links, is what we're going to spend a few minutes talking about. It's just different ways in which proteins attach cells to each other as you build tissues and organs. So the first intracellular junction or cell-cell junction we're going to talk about is called a tight junction. Its job, as it says here, is to prevent fluids and any of the molecules that would be dissolved in that fluid from passing in between two cells. So if we assume there's tight junctions here and we zoom in on that, it would look like this, where this is the cell membrane of this cell and this is the cell membrane of that cell. And what the tight junctions are doing is preventing anything from slipping in between the two cells here. So imagine like your stomach has acid in it. You want to make sure that none of your stomach juice, which contains acid, leaks in between your cells and starts dissolving the tissue that's underneath it. The analogy I use for tight junctions is like a Ziploc bag. Right? It is not sturdy and mechanical. You can easily open a Ziploc bag, but once it is closed, it makes an airtight or watertight seal, and that's what your tight junctions do. Then you have gap junctions, which our book does not have very good pictures of. So right here, this is supposed to be, let's say, the membrane of this cell here, and that is supposed to be the membrane of that cell there. And what the gap junctions are is basically a little tube that connects this cell to that cell. So over here, that red, highlighted in red, green in the middle, would be a gap junction. And primarily what gap junctions do is allow ions like sodium and potassium, but quite often our friend sodium, which is positively charged, it lets those ions pass from one cell directly into the next cell through those junctions. Because remember, ions have charge, they can't pass through the phospholipid bilayer, so there has to be some sort of protein structure that allow them to pass. Then you have desmosomes. These are strong mechanical junctions between tissues, or I should say between cells. And these are the junctions that hold your body together, that hold your tissues together. This is why you can pull up on your skin and it doesn't separate from the tissue underneath it. So let's use that as an example and say these are our skin cells here. That's not really what skin cells look like, but that's fine. Underneath your skin, underneath the epidermis, is a layer of connective tissue called the dermis. And all of the cells of your epidermis have desmosomes that attach them to the connective tissue so that when you pull up on your skin, you don't form blisters. It just pulls the skin and all the layers underneath it up with it. And if you look closely, the way they work is you've got proteins embedded in this membrane attached to proteins that are embedded in that membrane. So now we're looking at maybe this gap junction here. This is the cell membrane of this cell, and that is the cell membrane of that cell. And also then what you have, if I can draw, so if you look here, that is supposed to be parts of the cytoskeleton. You don't really know what that is, but it's just proteins that are inside of the cell. So these things extend out into the cytoplasm. So I'm just going to draw some extensions out into the cytoplasm here. and then these are going to attach to that. So if you imagine that there's desmosomes connecting all of these cells together, when you have sheets of cells like this, like the top of your skin, they're actually like row houses, like the brownstones, if you ever walk through Manhattan, or all the cool old historic row houses in Baltimore. Right. From the outside, each one of those houses has their own door. On the inside, they're separate from one another. But mechanically, structurally speaking, it's one big building that runs the length of the block. And what the desmosomes do, because they're connected to the cytoskeleton, which is then connected to the next desmosome, which is connected to the next cytoskeleton, it structurally ties all of those cells together. Again, so that when you pull up on your skin doesn't separate from the tissue underneath it, and your skin cells don't separate from each other. Speaking of cytoskeleton, that is what is next. These are 3 different cytoskeletal proteins. I don't care that you remember the names of them. The only ones that we're going to spend time talking about in this chapter is microtubules, because they make up your mitotic spindle. If you do look here, all of that in green are your microtubules as well. So they are present and important when the cell is not dividing, but also play an important role in dividing. And collectively, all of your different cytoskeletal elements combined with integral, or I should say membrane proteins, give the cell shape and structure, actually help it move. Cells can crawl along throughout your body, which is kind of cool to think about. And as we said here, they aid in cell division. So now we are on to transport, and back to sort of thinking about the phospholipid bilayer, what can pass through that, and then if things that can't pass through the phospholipid bilayer are going to pass through a cell, into or out of a cell, how are we going to deal with that? So we first break down membrane transport, and this means moving something either into the cell or out of the cell, into these 2 broad categories, passive transport and active transport. So as this says here, if the substance or molecule in question can pass through the membrane without any ATP being used, then that is passive transport. Any process that requires ATP expenditure is active transport. So we are going to have 4 different passive transport processes. First up is simple diffusion, which is when something crosses directly through the phospholipid bilayer. Then you have facilitated diffusion, and I changed this since I've posted it. I personally consider carriers and channels to be facilitated diffusion, and osmosis to be another form of simple diffusion. But what facilitated means, to back up for a second, is that there is a protein involved in the movement of the molecule, but that protein is not powered by ATP. And when it comes to osmosis, this you are going to see refers to the diffusion of water. Water can diffuse through the phospholipid bilayer, but there are also pores or proteins that water can diffuse through. So it could be either simple diffusion or facilitated diffusion when you're talking about water. So first we just have to define what diffusion is. And as this says right here, it is the movement of molecules from an area of high concentration to an area of low concentration. This is possible and doesn't require energy because molecules are always moving. This is what heat energy is the movement of molecules. Right? So if you heat up water, the water molecules start moving around really fast. If you get the water really hot, they move so fast that they fly apart from each other and become steam. So as long as you are above absolute 0 on the Kelvin temperature scale, which is negative, I want to say 296 degrees Celsius, which is really cold, right? As long as there's any latent heat at all in the environment, molecules are moving. When they move, they bump into each other, and when they bump into each other, they ricochet in opposite directions, which causes them to spread out. So when we say molecules move from an area of high concentration to an area of low concentration, we sometimes refer to that difference between high concentration and low concentration as a concentration gradient. And because molecules are moving from high to low, we say that molecules diffuse down their concentration gradient, right? And we've all seen examples of this. If somebody across the room from you puts on cologne or sprays some perfume, eventually those perfume molecules are going to make their way over to you. You're going to be able to smell it. That is diffusion, just molecules moving from where there's a lot of cologne originally, where it got sprayed, to all the way over to you, whether you want to smell it or not. So then, with simple diffusion, you have nonpolar molecules moving from an area of high concentration to an area of low concentration, passing directly through the membrane. This is how CO2 works, oxygen works, and water to a certain extent can also pass through the phospholipid bilayer. So water can diffuse. Next up is facilitated diffusion. As I said, this means a protein is getting involved. And there are two examples of facilitated diffusion: channels and carriers. First up is a channel. This is just, as it appears here, a simple tube or pore in the membrane that allows something to cross through the membrane from an area of high concentration to an area of low concentration. Gap junctions would be an example of a channel which let ions pass from one cell to the next. These are generally, if we're talking about a channel, they can be specific. So you can have a channel that just lets water through. You can have a channel that just lets sodium through or just lets potassium through. And then carriers are for things that are bigger than ions or bigger than water. And this picture doesn't really do it justice, but something like glucose, which would go through a channel, has, what does it have? 6 carbons, 12 hydrogens, and 6 oxygens. So it is like 12 or 13 times bigger than a single ion or a single atom. So for something like glucose, well, primarily glucose, but I'm sure there are other things. Glucose is the example that comes to mind. You need something like a carrier. And this picture doesn't really do it justice, but the way carriers work is that they're not open all the time. They're kind of like the revolving doors in hotels and stuff, right? There's always somewhat of a seal that the revolving door makes so that air doesn't blow in and out of the hotel lobby. With carriers, it's the same thing. It's always kind of pinched off, closed in the middle, so that anything smaller than glucose, like water and sodium and potassium, other things can't pass directly through it. It only lets specific molecules through and seals off the inside of the membrane from the outside of the membrane when the specific molecule it transports is not passing through it. And so now we are on to osmosis. Osmosis is just simply the diffusion of water across the cell membrane. The book, or I should say my PowerPoint, sometimes call it the plasma membrane. I try and get in the habit of just calling it the cell membrane because you have plasma in your bloodstream and the two terms are not related. So I just call it the cell membrane. And you do want to remember that water can pass through the phospholipid bilayer, but it can also pass through aquaporins. So your cells cannot prevent water from entering them or leaving them, but they can increase their permeability to water if there's a situation where a cell wants water to be able to pass through the pass through the cell membrane very quickly. Osmosis is very important to us because water is very important to us. So wherever the water goes, it has big changes on how things work. Okay, so I have changed things just a little bit. You have this slide. I did this one so I can draw a picture for you. So real quick, right, when we're talking about the diffusion of water, we mean the movement of water from an area where there's more water to where there's less water, right? Molecules move down their concentration gradient from areas of high concentration to areas of low concentration. Water does the same thing. It will move down its concentration gradient from an area of high concentration to an area of low concentration. And it's weird to think about the concentration of water, but you kind of have to. So let's imagine for a second, let me get a different color here. Yeah, that's good. That you have two glasses or two coffee mugs. One of the coffee mugs is empty. The other coffee mug has, oh, I don't know, let's say 18 tablespoons of sugar in it. Like it's almost full to the brim with sugar. Which of these coffee mugs am I going to be able to put more water into? The empty one or the one that's half full of sugar? You're probably thinking the empty one, right? So here you have two equal volumes of solution, right? If we fill both of these to the brim with water, this is all water. This one is going to be water plus a certain amount of sugar molecules. So there are more water molecules per unit of volume in the pure water glass than there are water molecules per unit of volume where there's sugar. So because solutes occupy space, they reduce the number of water molecules in a given volume, reducing the concentration of water in that compartment. And then osmosis happens when you have two watery solutions, like the inside of the cell and the outside of the cell, separated by a membrane, and there's differences in the solute concentration. This I forgot to point out. The total amount of all solutes is what is referred to as osmolarity. So in our previous example, we just had sugar in the water, but you could put salt and sugar and any number of other things in the water and it will dissolve. What determines how much room there is for water is the total amount of solutes in the water. So instead of talking about molarity, when you add up all of the solutes, it's osmolarity. And that's what determines which way the water is going to go. So here we have, imagine this is the outside of a cell, this is the inside of a cell. You have a semi-permeable membrane, which means some things can move through it, some things can't, right? If these are like sodium ions and sodium can't pass through your cell membrane, but water can, right? You've got a lot of sodium here, less sodium there, which means you've got a lot of water here and less water there. So water simply moves from an area of high concentration to an area of low concentration. So that's what's going on in this beaker. I skipped right ahead to talk about cells because that's what we care about. So because things like sodium and potassium and glucose, well, somewhat glucose, sodium and potassium and lots of other things don't always pass freely through the membrane, water does. So it is affected by the solute concentration inside of a cell and outside of a cell. So here we have three different examples. So we are going to assume that the osmolarity inside of the red blood cells is the same in beakers 1, 2, and 3. In beaker 1, they are referring to the solution as hypertonic. So hypertonic means it has more solute in it than whatever you're comparing it to. So in this example here, the solution is hypertonic to the cells, meaning there's higher osmolarity in the solution. If there's more solute in the solution, there's less water, meaning inside of these cells, it is more watery. There's more water molecules per unit volume inside the cell than outside the cell. So water is going to diffuse from inside the cells and outside the cells, and they're going to shrink up like that. When red blood cells do it, it's called crenellating. They form little things that look like crenellations on the top of a tube, or the top of a castle. Cannot speak. Here, the red blood cells are placed into what is called an isotonic solution. Isotonic means the same concentration or the same osmolarity. So if there's the same amount or the same concentration of solute inside the cell and outside the cell, then there's also the same concentration of water inside the cell and outside the cell. So you don't get any net movement, right? Water can move out, water can move in, but there's no gradient for water. So the volume inside the cell stays the same. If you put cells in a hypotonic solution, this now means that there is less solute in the solution per unit volume than inside the cell. So there's a higher concentration of water outside the cell, a lower concentration of water inside the cell. Water is going to move from an area of high concentration to an area of low concentration, and your cells are going to swell up. So you'll see in the future that because water can move into or out of cells down its osmolarity gradient or its concentration gradient whenever it wants to, your body puts a lot of time and effort into controlling where all of the solutes are. So if you can balance the solute concentration and make sure that all of your cells are in isotonic solutions, then you can balance the water movement and your cells don't shrink up or your cells don't swell and explode like these guys here. One of the other things you're going to see is that because your body can control where solutes go, one of the things that your body does is pump or move solutes from one compartment into another to get the water to move that way. So your body can't control water, so what it does is control the solutes and use that to set up osmolarity gradients and get water to diffuse from one bodily compartment into another. So now we're on to active transport. As I said, this is any process that uses ATP to move a solute across the plasma membrane. We're going to have primary active transport, or pumps, secondary active transport, and vesicular transport. Secondary we're not really going to spend a lot of time on. Vesicular transport we will talk about first, and I'm going to go all the way back to this picture here. If you have not had the lab yet, you may not know what a vesicle is. So any of these round organelles are just spheres of phospholipid bilayer with a specific fluid and proteins inside of them. So your cells have the ability to form little circular bubbles of phospholipid bilayer. Sometimes they're called organelles, sometimes they're just called vesicles. So if you imagine that there is a little bubble of membrane here and it makes its way over to the membrane, that vesicle can fuse with the plasma membrane and move something out of the cell, or you can use vesicles to move things into the cell. Let me go back and show you the picture then. So here we are. First up is endocytosis. So here you have to imagine that there's something big outside the cell that you want to bring inside the cell. So here you have your plasma membrane. Here's the thing that you want to bring inside the cell. So what the cell is going to do is surround that particle, and then this part of the membrane starts to invaginate and is going to move this way. And then these two things here are going to move out in that direction, and you're going to end up pulling or surrounding that particle with the membrane, and then that little part of the membrane pinches off and forms a vesicle. So this is regular endocytosis. This is called penocytosis, which we don't care about, as it says here, taking in particles that are dissolved in the fluid. And there's also what's called receptor-mediated endocytosis, which differs from this. In regular phagocytosis, or the sort of non-selective endocytosis, the cell is just surrounding something, whatever it is, and then bringing it into its environment. With receptor-mediated endocytosis, the cell can actually pick and choose what it wants to pull inside of it. So in this case, these stars stick to the green proteins on the surface of the cell. Then that region of the cell membrane is pulled inside and forms a little vesicle, and now those stars which were outside the cell are now inside the cell. So this is one way the cell can selectively pull large soluble molecules inside of the cell. And then exocytosis is just the same process in reverse. So here you have a vesicle. If you've had lab already, you should know that this vesicle came from the Golgi apparatus, because it is transporting something outside of the cell. So this vesicle is going to make its way over to the cell membrane. When this little bit of phospholipid bilayer comes into contact with your cell membrane phospholipid bilayer, the two fuse, and then what was inside the cell gets dumped outside the cell. So exocytosis is used a lot. This is how your neurotransmitters get released. Some hormones get released that way. Your protein hormones get released that way, and certain waste products. Let's see, we don't care about that slide. Let's skip it. Then we are into primary active transport, or pumps. So a pump is a molecule, or a protein, I should say, that uses ATP to move something across the membrane, generally speaking against its concentration gradient. That's the whole purpose of pumps, is if you're going to move something against its concentration gradient, that's like rolling a ball uphill, so you have to put energy into getting that thing to move if you want it to move from an area of low concentration to an area of high concentration. And that really should be on the slide up there. So if you haven't written this down anywhere, you are going up or against the gradient. That is the whole point of the pump, is to move things against or up their concentration gradient. So this is one specific pump in particular. It is the sodium-potassium pump, or the sodium-potassium ATPase. ACE because it's an enzyme and it uses the energy from ATP to move sodium and potassium. This is one of the most important molecules, phenomenon we are going to study, is the sodium-potassium ATPase. Every cell in your body has a sodium-potassium pump, some version of this pump here, and they all work pretty much the same way. Every time it burns an ATP, it takes 3 potassium and pumps them out of the cell, and then 2 sodium gets pumped into the cell. You are going to see in a few minutes that this is very important. And what it actually does is set up opposing gradients of sodium and potassium. So if you're moving the sodium out of the cell, you're moving it against its concentration gradient. So lots of sodium outside the cell, not so much sodium inside the cell. And because you're pumping the sodium in, you can generate a gradient and move the potassium, I hope I said potassium, from an area of low concentration to an area of high concentration. We are going to come back to this in just a minute. But we have to talk about cotransport first. Cotransport is when an energy from a gradient of one molecule is used to pump another molecule against its gradient. So here we go back to our friend sodium again. Because all of your cells have the sodium-potassium ATPase in them, then in every cell in the body, there's a lot of sodium outside the cell, not so much sodium inside the cell. This is a form of potential energy, right? It took energy to move all of this sodium, which means that gradient contains a certain amount of energy, which you can then use to power cotransport molecules and move other things against their concentration gradient. So now what you're doing is letting the sodium move where it wants to move, right? There's a lot of energy stored in this gradient, sodium molecules smashing into each other, and they want to move to an area of low concentration. So every time a sodium muscles its way into the cell, it pumps, in this case, an amino acid out. So again, this is the amino acid being pushed out of the cell every time the sodium pushes its way into the cell.